Okay, everybody. This is AP Bio Unit 5, Part 2. We're going to continue talking about meiosis. All right. Yesterday, did I get to this yesterday? Maybe, maybe not. Okay. All right. So, meiosis makes haploid gamete cells. And remember what a gamete cell is? That's either a sperm cell or an egg cell. And those can hook back up to go back to diploid. Okay. So meiosis results in daughter cells, and we're going to call sperm cells daughter cells, with half the number of chromosomes as the parent cell. So in humans, if I scooped one of your cells out of your skin or your muscle or wherever, it would have 46 chromosomes. If I went in and looked at the chromosomes in a sperm or egg cell, there would only be 23. So you'll go back to 46 when sperm and egg meet, and you go back to diploid. And so life kind of goes in a cycle that switches between haploid and diploid. Most of, the large majority of human life is diploid, okay? So the meiosis phase is very short for humans. To get to a haploid cell, you have to sort of do 2 rounds of division. And that's where we have meiosis 1 and meiosis 2. Okay, meiosis 1 and meiosis 2. All right, any questions on this slide here? We'll go ahead and move on here in a sec. I see people still writing, so I'm going to kind of hold off. Okay, go over there? Or wait 1 second. 1 second, okay. All right, we're going to go ahead and go forward. Before we go any farther, we're just going to compare these, okay? Which I think was one of the questions I asked for. I think this was one of the Wednesday questions. All right, so just to compare the differences, we already learned mitosis back in Unit 4 as part of the cell cycle. This occurs in somatic cells. Another word for somatic cell is body cell, okay? Another word for a somatic cell is a body cell. So all your body cells are growing and dividing, and that includes your first division after you're a sperm and egg meet up. So sperm plus egg equals zygote, and then everything else after that is mitosis. Okay, we get one division. Okay, the chromosomes copy, we divide them in half, we get one division, exact copies. Okay, the result are both cells are diploid. Both cells are diploid, and they are exact genetically identical. I combined 2 words and invented a new word, genetical. Okay, so they are genetically identical. Genetically identical. Now, in meiosis, we get gamete cells, not body cells, gamete cells. These cells divide in half 2 times, okay? So we're going to double it up, divide it, and we're going to go to half the amount, okay? So we get 4 cells that are haploid. So anytime you have sperm cells, they're sort of made in groups of 4, okay? They're made in groups of 4. Egg cells are the same way. In humans, we only use one of them normally. Okay, sometimes 2, and that's where you'll get things like twins. But most of the time, just one egg develops because we don't like humans are not the greatest at carrying multiple um, babies. Every single sperm and egg cell is genetically unique. This is why, again, you don't look like your siblings because these are all genetically unique. All right. I see my pencils and keyboards going. I'm going to kind of pause here for a minute for people to catch up. Okay, we're going to go ahead and move forward. All right, let's look at some of the big events in meiosis. Okay, there's three big events that are unique. Okay, so we've learned mitosis. Let's just dial into the things that make meiosis special. Okay, in prophase one, crossing over happens. This is where we have pieces of chromosomes that are going to detach from one another. Whoops. And sort of shuffle the deck on that chromosome. Okay. In metaphase one, tetrads, what's tetra mean? Anybody know what number that means? Four is right. Okay. Like tetrads, which are homologous pairs, line up at the metaphase plate. So tetrads. Okay. So sets of four. In anaphase one, the homologous pairs separate. Okay. They separate just like before. A equals away, so they do separate. All right. You guys getting this one in to your notes? Any questions on this? Um, Pretty much, yeah. I missed the word synapsis. Synapsis is where they sort of match up. Okay, so synapsis is where they match up. So if you're going to cross over chromosome one with chromosome one, the other chromosome one, the homologous chromosome one, synapsis is them coming together, and then crossover is when they actually start switching. So sorry about that. Synapsis is sort of pre-crossover. Okay, they've got to. You can't cross over unless you're sort of curled around each other. All right, was that too fast? No. Okay. So in interphase, the cell still goes through a G1, an S, and a G2. Okay, it's still going through that. So we start with one copy of each gene, and look, here's your tetrads. You have four of them. Now they're connected together like, I don't know if you can see, they're connected together like this. That's one, and that's one, so that's two of them together, but they line up in tetrads, so these little bundles of four. Okay, so these homologous pairs physically are connected to each other to make these tetrads. And this is where uh, crossing over happens. Now, this happens at an area called the chiasma. Okay. Chi is the Greek letter X. Okay. So you can kind of see that these are X. All right. So it's named for an X. I don't know why they couldn't just call it the X area. Yeah, I heard a chat come up. Can I go back? Yeah, let me go. I got a request to go back for just a sec. I went real fast. Okay. Here? Okay, let me park here for just a second. I sorry about that. All right, I'm going to go ahead back to where we were. So this chiasmata is this X-shaped area, okay? And DNA is exchanged between the homologous pairs at well-defined points, okay? There are sort of cut points where crossing over happens. If it was completely random, we would not end up with sort of new homologous pairs. So, because we have all this crossing over, each bundle of chromatid that is produced has a unique new combination of DNA. So all four of these are going to now be different, whereas before they were the same. Okay? So the purple one, let's just go with this purple one here on the left. Okay? There's two chromosomes there that are the same. Now they're going to be completely mixed up. The two yellow ones are going to be completely mixed up. How did that happen? Pieces of the purple one and pieces of the yellow one sort of shuffle together during the event we call what? Crossover is right. Okay. Now we have a new anaphase here. Okay. Now remember, go back real quick. It's okay. This is complex stuff. I need the feedback. That's the hardest thing about teaching. When you guys are all at home, I never know when to go forward. When you guys are in the room, I can look over and see if pencils and fingers are hitting keyboards, and I kind of like, okay, they're ready. But sometimes I miss that. How about now? Okay, I'm good. All right. So in meiosis one, it's a little bit different than mitosis. Okay. This is the metaphase. They're lining up in double in tetrads. Okay. They're not lining up as singles, they're lining up as doubles. Okay. So we call those tetrads. Um, but these tetrads have already gone through crossover. Hey, well, yeah. All right. So look at anaphase in meiosis one. Normally, in mitosis anaphase, got to get my pencil out here. Hopefully, I've got a little charge this morning. I do. And you tell me, what's Mr. Sater going to draw to explain this? Okay. What's different? What do you predict is going to be different about the anaphase? Okay, I'm going to go with blue. Normal anaphase, there are um, on those microtubules. There's one set. What do we have now? We've got the double set. So you kind of see like this double anaphase going on. Okay, this is only going to happen in meiosis one. Uh, in meiosis two, the anaphase is going to be singles. Okay, so that's this is a key way. Okay, this is a key way to know where you are. So if you ever see a picture and you're like, what there's doubles in there. Well, that's how you're going to know you're in meiosis one, because what's going to happen down the road is those are going to get cut in half again, and we're going to go back to haploid. So these pictures are a really big deal. You've got double, double, so those are your tetrads, and then you've sort of got these double anaphases happening, and then in meiosis two, it's going to be singles again, and those are going to be like setting up the haploid. So if you're going to divide twice to get the singles, you've got to have these doubles, okay? You can't do all this genetic variation and crossing over without doubling this. Okay, so homologous pairs of chromosomes. In mitosis, the big difference is it would not be pairs. Okay, so that would be the big difference. All right. Can you people at home see my slide, or am I sharing enough? Like, is it audio wise? You mean? Okay. Yeah, that can happen sometimes. Let me also share this. It might help a little bit. Okay. Let's delete this stuff and move forward. All right. So another big sort of clue or sign that you're in meiosis one is these are going to be still attached. Okay. At the kinetochore, they're still attached. Brian, did still attached circle? Okay. Good. All right. Ready? All right. We're good over there. Now, we're still in meiosis one. We're still in meiosis one. Okay. The big thing is we have two again. So telophase, T for two, is kind of like how I've been telling you to sort of keep that straightened out. So everything's pretty much the same right here, except for the number of chromosomes. Okay. Instead of single chromosomes or chromatids, we have still have uh, homologous chromosomes that have crossed over. That have crossed over. Okay. So these cells are like somatic cells, except that their DNA is what? It's been shuffled. They're not genetically identical. All right. Meiosis 2 looks just like mitosis. Okay. So these guys are going to prepare. They're going to line up. There's no crossing over in meiosis 2. Okay. That's already done. We're not going to do it again. And there's nothing to cross over with. Okay. There's nothing to cross over with. These guys, this is like chromosome number one, chromosome number two. They can't cross over. They don't carry the same information. All right, so we have, just like before, a spindle forms. Now, what's the difference here? They're not tetrads anymore. They're just homologous pairs. And these are going to do the same things. You're going to line up in the middle, metaphase middle, and they are going to be unique. Okay, they're their own separate entity. So if I go right down the middle of this and right down the middle of this, these are both unique chromosomes. So they're not genetically identical this time. You will get a lot of questions that are like, what's the difference between chromosomes in meiosis 2 of and what's in meiosis 1? So you got to kind of like pay attention to, they'll ask you that to see if you really know what's going on. All right. What's this going to be? These look like little letter A's to me. So this is anaphase 2. And we're down to four. We're getting ready to go down to four haploid cells. And here they are. Was that too quick? You guys aren't complaining. Okay, so these are either going to be four egg cells or four sperm cells. Okay, and go back for a minute. Yep, I'm sorry. Whoops. There we go. I'll park here for just a second. one of the reasons why I'm so like, hey, let me go forward is because I'm kind of like, in my mind, I'm like, well, these are the same. We don't need to spend any time on this. But I know you're looking at it as sort of someone who's seeing it maybe for the first time or the first time in a year or so. So I need to slow it down. All right. They're telling me they're good over there. All right. So now we're at that final stage where we have four haploid gametes. Okay. And how can we tell we're done? These cells have to be functional cells. Egg cells are some of the biggest cells in human anatomy. Sperm cells are pretty small. Um, and each one of these is genetically unique. Okay. So if these were eggs and you hooked up four genetically identical sperm cells, which doesn't happen, but if you did, each one of these would still be different from its siblings. Okay, so we're going to get lots of genetic variation. Two, you know, two, two, a male and a female coming together. If they stay together for life and have thousands and thousands of offspring, they would still be genetically unique. Okay, still be genetically unique. Now, maybe you're thinking I'm talking about humans, but I'm fish or something like that that lay lots of eggs. Each one is still genetically unique. Okay, there are exceptions to this. Okay, bees, a queen bee lays hundreds of eggs a day. All those are genetically similar. Okay, so there's always exceptions in biology. So every worker bee is exactly the same. They don't need genetic variation because worker bees don't reproduce. And they're all female too. All right. So just to review meiosis, okay? This is how you start and how you finish. The big thing you can see here is we're going from four, one, two, three, four, down to two, and these are all unique compared to the originals. Okay, so the DNA of these four daughters is totally different than the DNA of the parent. Those are the big things you need to see. Okay, this is going to be something that you're going to want to pay attention to because this is also a big theme for questions. Because when we get into evolution, genetic variation is very important. So, how does this work? Okay, we talked about crossing over. That kind of shuffles information on individual chromosomes. If crossing over didn't happen, certain groups of traits would never break up. They would always travel together and remain connected. So if we didn't have crossing over, let's just pretend two very visible traits were on the same gene. Let's say widow's peak and uh, blue eyes were on the same gene. That would mean if no crossing over happened, that would mean people with widow's peaks would always, always, always have blue eyes. And so because we shuffle these up, traits can kind of also get shuffled and bundles of traits don't travel together. So that leads to lots of variation. All right. The second factor, which people have a hard time understanding sometimes, is independent assortment. And all this means is when we say independent assortment, that means nothing is affecting the assortment. It happens randomly. So they are sorted independently And what does that mean? That means there's 23 pairs of chromosomes. One of them goes one way and one of them goes the other way. They don't all go one way and they don't all go the other way. They randomly go either way. Okay? So you're going to make four gametes, and the first pair is going to be divided up randomly. The second pair is going to be divided up randomly. The third pair, and so on. So it's not like all of the ones from one side go this. They they sort independently of the other 22. So that means that traits are mixing randomly again. So you have two randomization factors that lead to more variation. In a large population, variation is good. Okay? Some of the variation is going to be, doesn't matter. Okay? It's not like people with widow's peaks have a better chance of surviving and mating. Okay? But in an environment that has pressure, certain traits will be better than others. In humans, this is not as prevalent because not a lot of things are removing bad genes from the population. We have medical treatments that allow these genes to stay in the population. But if you're, a fish swimming out in the Scioto River, you better believe that this matters, okay? So populations will evolve based on the variations that are in them. If you have a very little amount of variation in your population, it can be very hard to survive changes. This is happening with endangered species, okay? You've probably heard like, this is the last white rhino in the world, okay? It's very hard to bring back a population from two or three individuals. It's even hard to do it from 50 individuals, okay? Because something in there will not have variation, and it'll be a negative trait that comes out over and over and over again. You see this with dog breeds, okay? What is it? Which dog breed gets the hip dysplasia? Is that no pugs? It's the nose thing. I thought it was labs, but I could be wrong. Their hips are out of alignment and their hip joints are loose. It's one of those things because a very small amount of founders of that breed had that problem, and that gene just shows up over and over and over again. Different breeds are known for different problems. Okay, so one of the healthiest dogs you can get is a crossbred dog where you got, oh, we're gonna bring in some mixed up genes, so those problems those negative genes that have been expressed over and over and over again get washed out. But people don't like that. They want their designer dogs and all that sort of thing. Okay. The next factor that allows for random um, uh, variation, I'm catching a little lag here. There it goes. Okay. Is that any sperm can fertilize any egg. So sperm and egg are recruited and join up randomly. A lot of people seem to think like, well, the strongest sperm has won. That's pretty random. Okay, it's not a factor. Okay, now there could be deformed sperm that leads to fertility problems, but in general, it's not like the strength of the sperm that are causing a lot of traits to be inherited. Okay, that's kind of like a cartoon image we get in our head that sperm are in this big sort of marathon race. They are, but it's not the first sperm that fertilizes the egg either. A couple early birds have to show up and start weakening the wall of the egg, and then they're sort of like offensive linemen, and then the quarterback sneaks through. That's the one who gets it. All right. Well, I mean, what you are the lucky one, but you weren't the first one. All right. So I'm going to kind of try to wrap up this whole lecture here. All right. The big thing you need to take away, yes, it's how meiosis works, but what's the point of it? Okay. The biological point of it is genetic diversity in sexually reproducing. Okay. Sex is expensive. Animals have to put themselves at huge amounts of risk. A lot of times, um, they put on these big displays. Cardinals don't blend in with the environment. Um, Animals will do crazy things energy wise to attract mates and put themselves at risk. You think of like male peacocks. They have this big giant tail, takes a lot of energy to grow. I imagine if you have a big tail like that, some bobcat or something out in the wilderness can grab a hold of you easier. Okay. They make a lot of noise to attract mates. So all these things are like I'm going to really put myself at risk to attract mates. Okay. Humans do crazy stuff like this too. Okay. I'm going to buy the most expensive car and I'm going to go out and, middle school boys are famous for this, like jumping up around and hitting signs on the ceiling and doing crazy things to be like, look, my genes are so intense, I can like destroy my body and still survive to mate with you. I don't know what's up with that, but it seems to be a big middle school boy thing to like hurt yourself in front of girls or something and kind of like show off your genetic strength. But we do a lot of work and we put in a lot of effort to attract the right kinds of mates. Okay. So it's not just animals that do this, it's also humans. Okay. So we put a lot of effort into this. Okay. A lot of people will joke around like the whole point of life is to put your DNA into someone else, your offspring. Okay. So I'm not even going to get into child rearing and how much energy that takes. All right. So this takes a lot of free energy from the environment, okay, to grow and to reproduce. So we talked about that. All this expense is worth it, not for the individual, for the population, okay? Now, breeding is not usually random in most biological systems. So, you watch the nature documentaries like the biggest, toughest lion is the one who gets to breed with everybody. That's not exactly accurate. There's a few sneaky ones on the side that work their way in, but in general, people are attracted to things that ensure better success of their offspring. Okay, and we can go into all that in humans and talk about waist to hip ratio. People think that's attractive. People think this is attractive. They're all supposed. Youth is attractive because people who are young are better at having babies. So I'm not going to go into every one of the factors that people think are attractive. Okay, and people think different things are attractive too. But in general, a lot of attraction features are related to um, offspring success or rearing of offspring. Okay. Most females are attracted to the big, strong guy. Okay. But there's other factors as well. All right. And thankfully, humans aren't driven only by those factors. There's other things we look at too, like is he a jerk or is he nice? Okay. Which again, is that so that you can have better offspring? I don't know. All right. This is my last slide. We're going to go ahead and stop our recording here.